Welcome to Living Machines. I'm Stephanie Lansing. Um, so we're going to talk about living machines. So what are living machines? They basically integrate principles from both ecology and engineering, and they utilize the natural abilities of organisms to break down and metabolize nutrients in wastewater, as well as carbon. So they rely on renewable energies, not exclusively, but they do reduce the amount of fossil fuels that you would use compared to traditional wastewater treatment. And normally they're going to be treating without the use of chemicals, flocculants, other things to assist in the wastewater treatment process. Their advantages, again, the idea of these aerobic anaerobic interfaces can help with the denitrification process and the degradation of carbon. Also, it's compartmentalized. So you often have these tank-based systems. If you're coming from one place, if it's a point source pollution, then you'll have different tanks that it goes through these tanks. But for non-point source pollution, you can even have these floating living machines that can deal with more non-point source pollution. So what have they treated in the past? So you'll see me use the word living machines, which is a copyrighted term. So you'll also see me use the word ecological treatment systems, so ETS. So in the past, whether it was called a living machine or an ecological treatment system, they have treated septic from septic tanks, municipal wastewater, dairy waste, as well as industrial wastewater. What they can create, in addition to clean water, you also have ornamental plants that are grown. You can actually, some of the sludge can be used as compost, and there's also been systems that have incorporated aquaculture. So what is this treatment process? So this is the components of the registered trademark living machines. And so first you have an anaerobic reactor. That is followed by an anoxic reactor. Again, with that anoxic reactor, we have some oxygen, but it's not anaerobic, it's not aerobic, it's very low oxygen concentration. And then here you have a closed aerobic reactor. That is followed in this system by 3 open reactors that have plants on them growing within the reactors. Then you have a clarifier. In that clarifier, you have settled solids. Those settled solids may go out as compost or go out for further treatment as sludge, or they may actually be reprocessed back to the beginning of the system if you need more organics back at the beginning of the system for denitrification. And then finally, you're going to have like an equalized fluidized bed. And that's some sort of kind of polishing system at the end. So we'll go through each of these one by one. So the anaerobic tank is designed to reduce that biochemical oxygen demand and solids via microbial degradation. And it provides a huge reduction in pathogens at that just very first step in aerobic tank. Basically, it receives that effluent and it acts as a settling tank. And then these settled solids are treated or reduced by microbial degradation. You can use like plastics or bioballs. Bioballs, you can think of them as being like a wiffle ball. So it has, the water can move through it, but it has a lot of surface area for microbes to attach to. And again, that accumulated sludge may be collected and treated, or it may just proceed through the system. And if there is any air emissions from this tank, you can pass it through a carbon biofilter for odor control if you're worried about the smell coming from the anaerobic tank. It then goes to an anoxic tank typically. Again, designs are all different, but in this kind of scenario, it would go through to an anoxic tank. And that basically is designed to not have anaerobic conditions, but really have a low oxygen concentration. So you want something less than 1 milligram per liter, but not 0, right? And basically you can do that by a coarse bubble diffuser. So basically you can think of it as like a PVC pipe with holes, big holes drilled in. So you have these big bubbles coming through. And those big bubbles actually provide oxygen, but also the reason why you want big bubbles is then they mix as well the water through that's being treated. The goal is to actually promote flock forming. And again, we're still going to get some denitrification in those conditions so that we can have removal of BOD and nitrate. Again, what's great about this tank is that we both have carbon still in there and microbes which they need for the denitrification process because they're heterotrophic organisms that use carbon as their energy source to reduce the nitrite to into gas. Again, this one we can also have some sort of media. So we can have biochords, which again is like a little fishing line that comes down that again provides that surface area for microorganisms. And again, it can also receive those biosolids from a clarification if we need supplemental organic matter for denitrification to occur. Then we might have a closed aerobic tank. Here we're going to have a highly turbid aerated environment using fine bubble diffusers. And we're going to increase the oxygen content. So that's going to aid in our BOD removal. And we're actually going to get nitrification. So this is where if we have ammonia coming in, this is really where we're going to see that reduction in ammonia into nitrate. It's designed to treat dissolved organics and the ammonia. And it also, that aerated environment is going to decrease any odors. Our planted tank, then, there's where we're going to get the majority of, again, of our nitrification and our BOD reduction. And instead of being covered with like kind of a biofilter on top as a closed aerobic tank, these are going to be covered with vegetation that are actually on top of racks. And the wastewater is actually going to flow underneath where all the roots are, that the roots are actually suspended in these tanks. And that root zone is going to provide surface area for microbial growth. The roots are going to uptake nitrogen and phosphorus. The roots, again, are going to provide habitat for beneficial insects and microorganisms. And then within those roots, when they get really thick, we might actually see anaerobic microcytes within there. So we might actually get denitrification occurring in those roots as well. In terms of plants, we want plants in these planted aerobic tanks that can take up nutrients at a high rate. Again, the population of bacteria and the denitrification that might occur in those microcytes is going to be related to the lability of the organic carbon that's leached from plants. So labile carbon is actually the fraction of organic carbon with a really rapid turnover time. And that's going to be a food source for those microorganisms. So again, if you might even have submergent or floating plant species, which may provide some of that labile carbon that's needed for denitrification, or you may have emergent species that come out at the top, and they may provide more like, twigs and leaves that are more recalcitrant. But again, that might help with long-term denitrification needs, slow release. You may even put in some decomposting twigs later in the system. If again, you still need denitrification to take place and you still need that labile carbon, then you still have nitrate that needs to be denitrified. Examples of some plants. So a submergent plant is where most of the structure of the plant's below water. Again, it can be subject to low light if it's still really dirty water. But again, they tend to be fast growing. So it might be a water weed or a pond weed, a water stargrass. You also might have floating plants. So those are ones that are not rooted down. And so those might be duckweed or coontail or bladderworth, or you're going to have emergent. So that's where the majority of the leaves and shoots and the flowering structure are going to be above the water. So that's going to be your cattails, your bulrushes, your wildlife, your sedges. So after the planted aerobic tanks, you're going to go to a settling tank where the settled solids they can be recycled back to an aerobic or anoxic tank. And that recycle loop again helps to select for beneficial microorganisms and increases the solid retention time, so we get more treatment within the system. Instead of just settling out the solids in a normal wastewater treatment system, you settle out the solids and then those solids are biosolids applied to a field. Here, by doing more recirculation, we can actually treat maybe a larger proportion of the solids that come into the system. Again, these clarifiers may be covered with duckweed to actually prevent growth of algae because we don't want to add too much more. And so decomposing, at this point, we're clarifying, so we don't want to add a lot more carbon at that point. And here we're going to get a large reduction in pathogens as well. And then finally, we're going to go to ecological fluidized beds or subsurface wetlands. And so this is kind of your polishing step where you remove your BOD, total suspended solids, and your nutrients. We may have crushed media to provide surface area for bacterial growth and to do that filtration process. And we also may use different types of media, so calcium or iron, to actually retain some of the phosphorus by absorption to that media that we have in there. We may actually also have, instead of it just being an anaerobic subsurface wetland, we may actually have an inner and outer tank, or we may actually have a vertical wetland where we have aerobic and anaerobic zones, so that we're again doing some of the following denitrification, nitrification that may need to happen. So I'm going to take you through an example. So this is the Waterman Ecological Treatment System, which was located at the Waterman Dairy Farm on the Ohio State University campus, and actually treated a portion of the wastewater produced from the milking parlor and 2 of the dairy barns. So the wastewater was actually diluted. It was very thick wastewater with 1 part wastewater to 3 parts well water. And then that wastewater was pulsed into the system every 10 minutes. And then it was distributed against 4 different treatment lines because it was a research project. So we had 4 lines in there to provide statistical replication. So here you have the dosing tank. It came into an anaerobic reactor, an anoxic reactor, a closed aerobic reactor, an open aerobic reactor with plants, a clarifier, which then circled back to the beginning. The solids did. The liquids continued on to a subsurface wetland, which then went to 2 more open planted reactors, another clarifier that then looped back into the first aerobic, the reactor 5, another aerobic reactor. And then the water then continued on through 2 subsurface wetlands before it was released. So here it is kind of inside the greenhouse. Again, all these things going in succession, but 4 lines that kind of take you through the system. But the waste came in again every 10 minutes and was distributed by a manifold into one of these 4 lines. And so here's the outside of the system. So here's the dosing tank where we filled it up with the wastewater and well water. And again, it was dosed into the system. And then here's the tank that held the effluent from the wetlands. So here's when the system was first built. So you can see here, we have planted systems. We have the tanks going through and the water pipes going from system to system. And you can see the subsurface wetland with crushed gravel and stone. And here, these are the clarifiers here. So this is when it's all just with water building the system. And here again, those mid wetlands before we went into more planted systems. And here's after 1 month of waste addition. So you can see the plants are now growing. They now have the nutrients. And this is after 3 months. So you can see large plant growth as it first comes in. And then we actually had more diverse plant growth as we move through the system. And then you can see the duckweed here on the clarifiers. So in the second half of the system, we actually planted the same amount of plants in both, and all the aerobic reactors had the same, we had a variety of plants that were seeded. And what happened was in the first half of the system, these rushes took over. Okay, so this is actually a papyrus plant and it took over. Well, in the second half of the system, we had a really nice diversity of plants that came. So as the nutrients got lower, we actually had a larger diversity of species than at the beginning of the system with very high nutrients, we had more of a monoculture. And so this is kind of the monoculture that you see that came in the first part of the system. Here are those biochords. So again, this is going to be in our anoxic tank. And so we have these biochords here that actually are, this may be the closed aerobic, that actually has surface area for, so these were white when they came in. They're actually like fishing line. And here's the plants. So you can see here in the plants, they provide surface area for microbial growth. And so we have, again, microbes and algae and all sorts of growth on those roots. And those roots are actually in the water. We're just lifting them up so you can see. And so here's what came into, this is with the 1 to 3 dilution of wastewater, which again, is complementary to what comes into a wastewater treatment plant. Again, they tend to get more solid separation. And then this is what came out of our effluent. So it was treated quite well. And so you can see that the reductions that we get, our total suspended solids coming in were about 430 milligrams per liter, and we had a 96% reduction. Our ammonia coming in was quite high because, again, this was manure, which has a very high ammonia concentration. So we didn't come in with hardly any nitrate. It all came in as ammonia. And we got 99% reduction of that ammonia. But we actually had a little bit more nitrate, but we're still way below regulation level, just because we were making nitrate in our process. Our phosphorus came in again fairly high, 21 milligrams per liter, and we got almost an 80% reduction in that. And then our BOD, our carbonaceous BOD, came in at over 500. And again, we got 99% reduction with less than 5 milligrams per liter in the effluent. And so if you look at these different, where they're kind of coming in, so this is a little bit hard to understand, but here's our ammonia and here's our BOD. So coming in, our ammonia is around 60 and our BOD is at this side. So almost over 600 milligrams per liter in this example. And so you can see the ammonia is going down. Even in our, these are our anoxic reactors. And then we get much more reduction in these aerobic reactors where, again, we're having nitrification that takes place. So our nitrate, which is our DO and our nitrate are right here, and they basically mirror each other. So when it's very low DO in the first 3 tanks, then we don't have a lot of nitrate. As the DO goes up, our nitrate is then going to go up as well. So our nitrate goes up and our ammonia goes down. And you see here, we don't get a lot of phosphorus reduction. And this is the green is the phosphorus in the first half of the system. It wasn't really until the second half of the system after that wetland. And then we have more planted plants here where we start to see that phosphorus really being reduced through uptake and through absorption, because again, a lot of the other carbon and nitrogen, especially the solids, they're being reduced as it goes through the system. And then finally, by the time we get to our effluent, we're close to 0 with everything, but our DO is still high, our phosphorus is still a little elevated. But what was interesting, this is just looking at the phosphorus. The phosphorus varied by line. So again, we had 4 lines that received the exact same thing. And yet what we found was in the different lines, so here's one line with the phosphorus kind of going up and down and then being reduced. But what we found was over time, this is the effluent phosphorus. So at week 1, it's pretty low. But over time, some of our systems actually went up, some of them went up and then went back down. And what we found is that in our final wetlands where we were getting a lot of our phosphorus removal, some of them had a preferential flow pattern. And so we were having some of the phosphorus in some of the wetlands where we could kind of see breaks in the wetland where the water was moving through. So you really need to think about how that wetland flows and making sure that you're using all of the surface area of the wetland for treatment. Again, this just shows 2 different lines, and you can see they behave differently, which is why you have that repetition. So we had, again, aerobic reactors here, and we can see a huge decrease in the ammonia in line 2 in October from these aerobic reactors. And then by the end, it was just polishing, they were done. While this system didn't see a lot of reduction there. And so we really needed that second half of the system. So again, they're biological, ecological systems with microbes and plants interacting. And so they may not behave in the way that you think they're going to behave. And even though 2 systems are seeded the same, they may actually have different inputs, which is why this kind of idea of having repetitions of this aerobic, anaerobic, and plants and interactions, so that we can have that consistency at the end, because we know these are biological systems. This is another example of a system here at the University of Maryland, where we had wastewater coming in and then we had a trickling filter. So the only use of pumps really was we pumped the water up to this bioball trickling filter system and had that kind of giving our aerobic zone. And then it went to, and then it was anaerobic in this tank below. And so aerobic, anaerobic, aerobic, anaerobic. And then it went through these different vegetation tanks where we had hyacinth on top of them. And so this ecomycene was a sequential treatment system with a recirculating trickling filter and a series of these 3 aquatic tanks. The volume of the system was about 200 gallons and the retention time is about 21 days. It was loaded with simulated sludge, which was actually made from commercial dog food mixed up with water to get the right concentration of carbon coming in. and it reduced that chemical oxygen coming in from 3,500 milligrams per liter to less than 500. So we still had some elevated, but it was a great reduction. And so here's kind of the system. Here's kind of the anaerobic wastewater, and here's our trickling filter where it pumped up and then went down through this aerobic zone. And you can see here our water hyacinth. We can see here that sometimes the water hyacinth was looking pretty healthy. Sometimes we got a little ammonia burning in there from too much nitrogen, but it was all housed within a greenhouse on the back of the environmental science building at the University of Maryland. And so again, you can see here, the phosphorus kind of went up and down, but it generally, the trickling filter, it went down, and then we had some phosphorus, residual phosphorus in the tanks. But again, our dissolved oxygen just pumping through that trickling filter with the bioballs and all the microbes on it, we were able to have a very high dissolved oxygen level starting at 1 and going up to where we want to be around 7 or 8 milligrams per liter dissolved oxygen. So when we're thinking about designing living machines, there's a lot of things we need to think about. And number one, this is from Todd and Todd, their 9 design principles for living machines. And their number one thing they want you to focus on is the microbes. The microbial community is so important. Number 2 is the photosynthetic community. So there's our plant communities. We really need to focus on what plants we're using and how we're, in those communities and what types of plants and the diversity of plants. Again, number 3 is the linked ecosystem and the law of the minimum. And so what this means is we need to link these different systems together. So whatever one system is missing, we can bring that and recycle that back through. And so that we actually address these limiting nutrients and limiting agents by using recirculation and by using linked systems from one to another. This idea of this pulse exchange, again, if you have a pulse system, the idea is it'll work at a higher efficiency. We need to have nutrient and micronutrient reservoirs. So there's places where we were thinking about labile carbon, and we are thinking about the phosphorus that might be needed for plant growth, or the nitrogen. And so again, all of these, the nitrogen phosphorus ratio, all of these things work in conjunction. So we need to think about where we might have those reservoirs in the system. We need to think about geological diversity and mineral complexity. Again, these microbes, these plants, they need these minerals. And again, we're talking about absorption and removal. The types of minerals that we have in there are very important for these processes to occur. We need to think about steep gradients. So again, going from here to here to here to here, that's where we're getting a lot of our removal, is in those steep gradients, where it's going from aerobic to anaerobic back to anaerobic. And so that we can get those different environments, and we can have different types of microbes throughout the entire system. We also need to think about phylogenetic diversity. So here we're talking about not just microbes, not just plants, but all the phylas. Can we have snails in there? Can we have fish in there? Can we have different systems in there? And thinking about the diversity of ecosystems, how can we put that into the living machine? And then finally, that the microcosm should be a mirror of the macrocosms. So we're creating little ecosystems in these tanks, and how are those going to actually reflect what we would see in the natural environment? And how can we design something to reflect it as close as possible? All right, thank you very much.